All right, let's dive in. Today we're talking about one of biology's most incredible feats of engineering, a solution to a storage problem that seems, well, basically impossible. We're going to explore the absolutely brilliant way our cells package our DNA.
Just let that sink in for a second. If you took the DNA out of just one single cell and stretched it all the way out, it would be almost 2 meters long. That's taller than most people. So how in the world does all of that get crammed into a nucleus that's thousands of times smaller than the dot on this eye? The math just doesn't seem to add up.
But of course, nature solved this packaging problem ages ago with a truly elegant piece of biological machinery. And the secret? It all comes down to a structure called the nucleosome.
At its core, the strategy is something you already get, right? If you have a super long piece of thread or a garden hose, you don't just shove it in a box. You wind it around a spool. It keeps it neat, compact, and, most importantly, it doesn't get tangled. Well, our cells do pretty much the exact same thing with our DNA.
Okay, so if our DNA is the thread, then what's the spool? The answer is a special group of proteins called histones. And really, understanding these little guys is the key to figuring out this whole system.
So the spool's core is built from 4 main types of these histone proteins. But here's what's just wild. These proteins have barely changed over 1 billion years of evolution. I mean, a histone from you or me is practically identical to one from a simple baker's yeast. That's a huge clue, right? It tells you that their job is so absolutely fundamental to life that nature perfected the design way back when and has basically said, yep, don't touch it.
And the real secret to their function is this very specific shape. It's called the histone fold. You can kind of think of it like a universal Lego piece. This unique structure lets each histone protein know exactly how to snap together with the others. It ensures that the spool, this core, gets built perfectly every single time.
So we've got our protein building blocks. Now let's see how the cell actually puts them all together. And this is no accident, no random clumping. It's a very precise, step-by-step assembly line.
The whole thing builds from the inside out. First, 2 of the H3 and H4 histones pair up, and then that pair grabs another H3-H4 pair. That forms a really stable 4-part foundation. Then, this central piece recruits 2 of the H2A, H2B pairs, which kind of lock onto each side. And there you have it, a perfect 8-protein core. That's our histone octamer.
Okay, our protein spool is fully assembled. It's time for the main event, wrapping the DNA around it. And the level of precision here is it's remarkable.
The DNA doesn't just loop around randomly. Nope, it makes exactly, get this, 1 2/3 turns. That's 1.65 times around that histone core. And this specific left-handed coil is the fundamental unit, the basic building block of all DNA compaction.
So how does it stay put? This number is just incredible. There are 142 hydrogen bonds acting like tiny little magnets, anchoring that DNA right to the histone core. That massive network of bonds makes the whole package incredibly stable and secure.
Now here's the really clever part about how it works. About half of those 142 bonds are formed with the DNA's sugar-phosphate backbone, not the specific A's, T's, C's, and G's of the genetic code. And that's the genius of it. Because it's a universal interaction, it doesn't matter what the gene sequence is. The cell can grab and package any stretch of DNA it needs to using this exact same system.
But this is where the story gets even more sophisticated. You see, the nucleosome isn't just about static storage, like putting something in a box and forgetting about it. No, it's a dynamic system that actually helps control which genes get turned on and off.
So, sticking out from each of those 8 histones in the core is a long, flexible tail. You can think of these tails like little communication antennas, just poking out from the main structure, ready to send and receive signals from the rest of the cell.
And this line from the source material really hits the nail on the head. These chemical modifications are basically a code. The cell can stick different chemical tags onto these tails, and that can signal the DNA to wind up even tighter, hiding a gene away, or to loosen up, making a gene accessible so it can be read. It's an entire control panel built right into the packaging itself. It's amazing.
And the cell can customize its spools even more. On top of the standard histones it uses for general, stable packaging, it also makes these specialized variant histones. It can swap these in to create, for example, a less stable spool. And why would it want to do that? Well, it's perfect for genes that need to be unwrapped and read over and over again, really quickly.
So we end up with this really profound idea. We tend to focus so much on the genetic code, the sequence of letters, but the nucleosome shows us that the physical packaging, the shape and the accessibility of our DNA is just as crucial. It really makes you wonder, right? What if how we package our genes is every bit as important as the genes themselves? Thanks for joining this explainer.